So plasma membrane. Plasma membrane, what does a membrane do? Membrane, it forms boundary both around the cell and around subcellular compartments like nucleus, mitochondria, lysosomes, which I'll describe in my next tutorials. So first of all, plasma membrane has three kinds of lipids. It is made up of lipids and proteins also. So lipids, they constitute 42% of the total composition of the plasma membrane, and among this 42%, phospholipid contributes 25%, cholesterol 13%, and other lipids constitute about 4%. This is made up of protein. It is selectively permeable membrane and it forms the external boundary of the cell. Now let's talk about, first of all, I'll describe this property, selectively permeable membrane. What does it mean? Selectively permeable, suppose this is the structure of a membrane. Membrane is having small pores in it, and so this is the inside and this is outside of the cell. Suppose there are some molecules which are of this shape. So if the cell requires this molecule, so it will simply go inside the cell if it is of the size of the pore. But if it is bigger in size, then it will go by the process of endocytosis. I hope you all are aware of these endocytosis, phagocytosis, exocytosis. If you are not, then I'll describe all these things in my next tutorials. So as of now, you can remember that if this molecule is of the bigger size, then it will go inside through the process of endocytosis, right? But suppose there is a molecule which can cross this membrane easily, but it is not required by the cell. So kya hoga? This will not let it cross through the membrane. This plasma membrane will prevent this molecule from going inside. Okay, so you might have heard about semi-permeable and selectively permeable. So what is the difference between these two, semi-permeable and selectively permeable? Guys, when this membrane is a part of the living system, we'll call it selectively permeable. But if we take out this membrane from the living system and put it in a beaker, then it will lose this property. What property? It will not be able to check the passage of molecule through it, which is not required by the cell. Moreover, यहाँ पे phagocytosis वगैरह process, endocytosis will not happen. So this is semi-permeable. जो इसके size का जो molecule होगा, that molecule will be able to cross this membrane, right? So this is the difference between semi-permeable and selective permeable. When this membrane is a part of biological membrane, living system, not biological membrane, but when this membrane is a part of living system, then we will call it selectively permeable, because it has the ability to check the passage of molecule which is not required by the cell. But if we take out the cell from this living system and put it in a beaker for research purpose or whatever purpose it is, then it will not have the ability to check the passage of the molecule which is not required by the cell, even if it is of its size. And phagocytosis, endocytosis, these processes will not take place when it is a semi-permeable membrane. So this is the difference between these two. Moreover, how this process, ये endocytosis के लिए जो pouch बन रहा है, how is this thing happening? So this is because our membrane is fluid, it's not static. Membrane is having a property which is called fluidity of the membrane, which enables it to form these pouches. So ये zigzag undulating movement में रहती है, right? So this process is possible because our membrane is fluid, it's not rigid. So now let's talk about lipids and proteins. Lipids, phospholipids. As you can see, it's having hydrophilic head and it is having two hydrophobic fatty acid chains. So basically they have four components: fatty acid, these two chains, as you can see, then glycerol backbone, and they are having phosphate, and phosphate is attached to an alcohol. Okay, these I have made wrong, sorry. This is the hydrophobic part of the phospholipid, and this part is hydrophilic. So phospholipids, they are amphipathic in nature. There are three major types of lipids: the glycerophospholipids, sphingolipids, and sterols. Now glycerophospholipids. Glycerophospholipids, they are made up of three components: a phosphorylated head group, a glycerol backbone, and hydrocarbon fatty acid chains. As you can see here, phosphorylated head group, this is phosphorylated, this alcohol, this is attached to a phosphate, so this is phosphorylated head group, right? And this phosphorylated head group is attached to the glycerol backbone, and these are having hydrophobic fatty acid chains. So this is the basic structure, which is called phosphatidate. It's virtually absent. So you can see यहाँ पे, this is the glycerol backbone. This is the glycerol backbone. Here you can see phosphate is attached to this glycerol backbone, and it is having two fatty acid chains. Later on, this when serine is attached to this phosphate, then it becomes phosphatidylserine, right? Major glycerophospholipids are phosphatidylcholine, phosphatidylethanolamine, it's inositol, serine. Glycerophospholipids are the most abundant phospholipid molecules found in plasma membrane, right? So in this case, I have shown that it is phosphatidylserine because serine is getting attached to this phosphate head group. If अगर यहाँ पे serine नहीं attached होता, if it is choline, then we will call it phosphatidylcholine. If it is ethanolamine, then we will call it phosphatidylethanolamine, right? If it is inositol, then we will call it phosphatidylinositol. To describe this structure, this phosphatidate, which itself is present in small amounts in membranes, I'll say that this is carbon number 1, this is carbon number 2, and this is carbon number 3. So this is the simplest glycerophospholipid, phosphatidate, which is also called as diacyl glycerol 3-phosphate, which has only a phosphoric acid group esterified to carbon number 3 of the glycerol, right? Major glycerophospholipids like phosphatidylserine, phosphatidylcholine, inositol, they are derived from this phosphatidate. In these other lipids, like this phosphatidylserine, this phosphate is further esterified to the hydroxyl group CH2OH, यहाँ पे CH2OH होगा. So this is esterified to the hydroxyl group of one of several alcohols like choline, serine, ethanolamine, inositol. Basically, membrane lipids, they contain phosphate group, which makes them phospholipids, because most membrane proteins are built on a glycerol backbone. They are called phosphoglycerides, unlike triglycerides, which have 3 fatty acids instead of 2 in case of phospholipids, phosphoglycerides. And moreover, triglycerides are not amphipathic in nature, whereas phosphoglycerides, they are amphipathic in nature. So this phosphate group is negatively charged, polar end, which is hydrophilic. Fatty acid chains are uncharged, non-polar tails, which are hydrophobic. And this glycerol, this is also hydrophilic. Question should come to you that how does a shampoo or a soap works? Shampoo or a soap works. How does it work? So they are made up of amphipathic molecules, right? It means they are having both hydrophobic regions and hydrophilic regions, right? This is the meaning of amphipathic nature. So their polar section will combine with water. Polar section will combine with water, and their non-polar section, which is hydrophobic, will stick with non-polar molecule like grease or oil, right? So suppose in normal case, if we are putting water to the grease or to the oil, it will not interact with water. Why? Because it is hydrophobic in nature. Oil or grease, they are hydrophobic in nature, so they will not interact with water. This is simple. Now when you are adding soap or detergent or shampoo to the water, it's going to have both hydrophilic and hydrophobic parts. So hydrophobic region will interact with the oil or grease, and hydrophilic part will interact with water. So what's next? After this interaction, this soap molecule will take the grease with them. It will take away the grease. Now let's talk about sphingolipid myelin. This is the commonest sphingolipid. They are having sphingosine backbone, whereas in case of glycerophospholipids, they are having glycerol backbone. A fatty acid, basically they have two fatty acids. One is coming out of this backbone, sphingosine backbone, and one is attached to this sphingosine backbone, just like the glycerophospholipids, where two fatty acid chains are attached to the glycerol backbone. They are having a phosphate also and an alcohol attached to the phosphate. In sphingolipids, amino group of the sphingosine backbone is linked to a fatty acid by an amide bond. Amide bond is basically C double bond O NH2. So here you can say this fatty acid, this is the amide bond, okay? This is going to be the amide bond. So you can see this fatty acid is getting attached to the sphingosine backbone. Here you can see the presence of phosphate, and this is sphingomyelin, where alcohol is getting attached to the phosphate of sphingosine backbone. These sphingolipids, they are less abundant class of membrane lipids, and they are derivatives of sphingosine. Sphingosine is basically an amino alcohol that contains a long hydrocarbon chain. And ये जो structure है, this sphingosine and fatty acid, this is called ceramide. Okay, ceramide is composed of sphingosine and a fatty acid. Now this will help in making up of sphingomyelin. This helps in programmed cell death, its differentiation and proliferation. To wrap up the thing, I can say that sphingomyelin is the most common sphingolipid. It's having a sphingosine backbone in place of the glycerol in glycerophospholipids. Like the glycerophospholipids, they have a phosphorylated head group, either choline or ethynyl amine. If it is phosphoryl choline, you can see. So we are calling it sphingomyelin, right? Now glycosphingolipids such as cerebrosides and gangliosides. So in case of cerebroside, only one sugar residue will be attached to the sphingosine backbone, and in case of gangliosides, more than one sugar residues will be attached, right? Galactocerebroside is one of the most simplest glycosphingolipids. It's having only a single galactose residue attached to the ceramide, and if you remember, ceramide is sphingosine plus this fatty acid. This is ceramide, okay? So in case of galactocerebroside, galactose will be attached to the ceramide. First glycolipid to be identified as a key antigen in experimental autoimmune neuritis. Now ganglioside, they have several sugar residues including at least one sialic acid. They are gangliosides, major constituent of most mammalian plasma membrane. They are present in abundance in the brain cells. So our nervous system is particularly rich in these glycosphingolipids or glycolipids, we can say. Myelin sheath contain galactocerebrosides. Now let's talk about sterols. The sterile cholesterol is major constituent of animal plasma membrane. It's absent from prokaryotes. It's made up of fused ring system. Its structure is of fused ring system. It's precursor of steroid hormones. Plants are having sterols in the form of stigmasterol and beta-sterol. I'll talk about this cholesterol in detail in my coming tutorials. This cholesterol is very much important in our living system, in our plasma membrane. In certain animal cells, they constitute, this cholesterol makes up about 50% of the lipid molecule in the plasma membrane. So it holds a great importance in the plasma membrane. So I'll talk about this cholesterol in detail in my next video. So this is the structure of our plasma membrane. So as you guys know that empathic molecules, these lipids, these phospholipids, sphingolipids, they are empathic molecules. So when we are putting this empathic molecule in an aqueous solution, these empathic molecules will orient themselves in such a way that prevent these hydrophobic regions coming into contact with the water molecule which is present outside the cell, right? So what has happened in the case of soap which is having a, suppose sodium palmitate. Sodium palmitate, it's a constituent of a soap which is having a single fatty acid chain. So when you are putting this into water, so it will form micellar, right? This structure. But in normal case, suppose these are having two fatty acids. Normally lipids, these are having two fatty acid chains. So it will become too bulky. It's not possible to fit these two fatty acid chains in a micellar like structure, right? So because of this, to avoid this bulkiness, it avoids to form a structure like this, and it forms a lipid bilayer, this sort of a structure. The most favored structure for most phospholipids in aqueous solution is a two-dimensional bimolecular sheet or lipid bilayer. These lipid bilayers, they will spontaneously self-assemble in aqueous solution, and the major driving force behind this is the hydrophobic effect. These hydrophobic fatty acid chains avoid coming into contact with water because they are present interior, they are present in, they are facing each other, they are in the hydrophobic region, right? So once formed, the bilayer structure is maintained by multiple non-covalent interactions, including this hydrophobic interaction they are having with each other, and van der Waals forces between the hydrocarbon chains, right? And what else? Hydrogen bonding between the polar head groups and the aqueous solution, right? These are the multiple forces which helps them in maintaining their shape, their size, and their fluidity. About the plasma membrane, in my next video I'll talk about the plasma membrane in detail.